You settle into the quiet of your own body, letting the world's noise fade into the background. There is a rhythm here, a rhythm so subtle you almost miss it if you are not listening closely. It is the quiet orchestration of life itself, each cell vibrating with purpose, each tiny organelle performing a task choreographed over millions of years. As you breathe, inhale and exhale, you feel the soft pulse of life, a gentle reminder that you are a vessel of countless wonders. Imagine a river, wide and winding, glimmering with the reflection of stars not yet born. This is your DNA, a flowing strand of possibility. It twists upon itself in spirals, a double helix coiling like a staircase made of light. Each rung, a pair of nucleotides, holds a code, a whisper of who you are, who you might become. You sense it not as information, but as music, soft, undulating notes that rise and fall in a rhythm only your deepest self can hear. Within every cell, tiny molecular machines work in harmony. Enzymes glide along strands of DNA, reading, copying, repairing. Proteins fold in intricate patterns, like origami made of sunlight and shadow. Lipids and sugars arrange themselves into membranes, crafting boundaries that define yet connect. You imagine them as dancers moving across a stage, each step precise, each movement essential. You feel a quiet awe as you recognize that all this choreography is happening within you, beneath the awareness of thought, in a world invisible yet vividly alive. If you pause long enough, you can hear it, a hum, a low vibration that threads through every cell. It is the hum of metabolism, of life converting energy into motion, into thought, into feeling. This hum is not loud or urgent, it is a soft, constant affirmation that you exist, that life persists. You let your mind drift along this vibration, feeling each cell's gentle murmur as part of a greater song, a lullaby composed over eons. The nucleus, a tiny command center, houses your genetic library. It is a quiet sanctuary where your DNA curls like ancient parchment, inked with sequences that hold memories of ancestors you never knew. Each gene is a phrase, each regulatory sequence a pause, a rhythm that guides how life unfolds. You can almost see them as lines of poetry written in a language older than time. The story is yours, yet shared with every living thing, a universal manuscript that connects you to the vast network of existence. Now, drift your attention to the mitochondria, the power plants of your cells. Here, chemical energy transforms into life's motion. Glucose and oxygen dance together in a waltz of electrons and protons, producing the ATP that fuels every heartbeat, every blink, every thought. You feel your body's energy flow like a gentle river, surging and subsiding with your breathing. It is humbling to consider that these tiny structures, invisible to the eye, are responsible for the spark that animates your entire being. Proteins, too, deserve your quiet attention. They fold and twist, adopting shapes that allow them to perform countless functions. Some carry messages, whispering instructions from one cell to another. Others build, break, or defend, protecting the delicate balance of life within you. Imagine them as artisans in a sunlit studio, shaping your existence with each deliberate motion. Their beauty is not just in what they do, but in how they do it, with precision and grace that defies comprehension. As your awareness widens, you notice the spaces between molecules, the invisible scaffolding of water and ions. They form the medium through which energy and information flow. Even emptiness is not empty here, it is charged with possibility. You feel the subtle push and pull of these forces, the delicate balance that allows cells to communicate, to adapt, to survive. This invisible world is a reminder that life exists not only in substance but in connection, in movement, in potential. You imagine walking through this landscape, a universe within yourself. Each cell is a galaxy, each organelle a planet, each molecule a star. The rhythm of life pulses through this cosmos, quiet but insistent, and you are both observer and participant. There is no rush, no urgency, there is only this continuous unfolding, a narrative written in the language of chemistry and light. You feel a deep, meditative calm as you float among these inner worlds, witnessing the poetry of existence. Your thoughts drift to the concept of replication, the quiet miracle occurring in every moment. DNA copies itself with remarkable fidelity, ensuring continuity through generations. Mistakes happen rarely, and when they do, life adapts, evolves, and grows. You sense in this a metaphor for your own journey, the balance of persistence and change, of stability and transformation. It is a rhythm mirrored in the heartbeat, in the rising and falling of breath, in the soft undulations of consciousness itself. Consider the epigenetic whispers, the subtle markers that influence which genes awaken and which remain silent. These are not rigid rules but gentle suggestions, tuning your genetic symphony according to experience and environment. You feel a kinship with this process, recognizing that your life, like your cells, is guided by both inherited instructions and present choices. The harmony is ongoing, a dialogue between history and now, a soft, unfolding story without finality. As you continue this inner journey, notice the gentle movements of the cytoskeleton, the cell scaffolding. Microtubules and filaments extend like delicate threads, supporting structure, enabling transport, guiding motion. They are silent architects, shaping the interior landscapes of your cells. You feel the subtle interplay of structure and flexibility, realizing that even in microscopic worlds, balance is everything. There is a quiet lesson here, that stability allows motion, and motion sustains life. You linger for a moment on the wonder of sensory perception at the cellular level. Receptors on membranes detect signals, responding to light, chemicals, and pressure. Every sensation you experience is first filtered through these microscopic sensors, converted into messages that your mind can understand. You imagine the flow of information like a gentle river, carrying whispers from the external world into the sanctuary of your awareness. This connection between inner and outer worlds feels tender, intimate, and profoundly alive. Finally, you let your attention rest on the interconnectedness of all these processes. Each cell is not isolated, it communicates, collaborates, and synchronizes with neighbors. Signals pass like gentle currents, harmonizing rhythms, ensuring cohesion. You sense a larger pattern, a quiet intelligence that emerges not from a single point, but from the collaboration of countless, tiny agents of life. It is a soft, invisible web, linking you to the rest of your body, to every living being, and to the world beyond. And in this reflective quiet, you feel the gentle hum of existence, a music composed of molecules, energy, and light. It is a song without words, yet profoundly understood. You drift in the knowledge that life is both delicate and resilient, intricate yet simple in its foundational patterns. The story of your cells is not separate from the story of the universe, it is a reflection of the same poetry that shapes stars, rivers, and the gentle unfolding of time itself. You exhale slowly, imagining your own consciousness as part of this vast, internal cosmos. Each heartbeat is a soft echo of creation, each breath a reminder of continuity. You sense the invisible connections, the subtle flows, the delicate balances that sustain all living things. And as you feel the whispering of your cellular universe, you notice a question beginning to form in your mind, a curiosity that beckons you to explore not just the blueprint of life, but the forces that shaped it across the eons. You settle into a soft light, the world of cells and molecules stretching around you like a quiet forest of infinite detail. Each leaf, each branch, is a strand of DNA, coiling and uncoiling with a rhythm all its own. Here, the language of life is written in letters you cannot see, yet somehow feel. It is not chaotic, it hums with purpose, a hidden symphony that has played for billions of years. And today, you are invited to watch one of its most delicate dances. CRISPR enters like a whisper, a tool both ancient in inspiration and new in practice. It is not a hammer or a knife, but a gentle hand that can touch and guide the flow of life's instructions. You imagine it as a tiny, luminous figure gliding along a DNA strand, scanning, pausing, recognizing. When it finds the sequence it seeks, there is no violence, no abruptness, only a quiet, precise snip, like the gentle trimming of a bonsai tree, each cut intentional and considered. You breathe slowly, matching the rhythm of the CRISPR as it moves. There is a grace here that surprises you. It is a dance of recognition, a conversation between the tool and the code it touches. Each snip is followed by a pause, a moment of thought, allowing the DNA to respond, to realign, to continue its endless spiral with subtle adjustment. This is not control imposed from the outside, it is guidance, a co-creation between human ingenuity and nature's own design. As you watch, you notice the surrounding molecules, the enzymes, the scaffolds that support this delicate choreography. They shimmer with energy, small sparks that mark interactions invisible to the naked eye. CRISPR moves in harmony with them, like a figure skater gliding on an invisible ice, every movement balanced by unseen forces. The dance is precise, yet full of possibility. You realize that with each careful intervention, the potential for healing, for understanding, expands quietly before you. You think about the stories written in DNA, the whispers of generations past, the patterns that define the cells of your own body. To step into this world is to touch something deeply personal, even if only metaphorically. CRISPR does not erase these stories, it reads them, gently suggesting edits where nature or circumstance allows. It is a conversation, not a command. You feel the weight of responsibility, but also the awe of collaboration, a reminder that science, when approached with respect, can be an act of listening as much as an act of doing. The process feels meditative. Each motion, each cut, is deliberate. There is a rhythm that mirrors your own heartbeat, subtle yet persistent, a steady assurance that change need not be abrupt or forceful to be effective. You imagine a strand of DNA, long and winding, like a river stretching across time. CRISPR moves along its banks, smoothing rough edges, gently redirecting flows, allowing the water to find its own course. You sense that even the smallest change here could ripple outward, transforming the landscape in ways both visible and invisible. Your mind drifts to the questions this tool evokes. What is possible when life can be edited with such care? What does it mean to intervene without breaking the harmony of what already exists? These are not questions with immediate answers, but rather gentle invitations to wonder. You feel the curiosity rise in your chest, not like urgency, but like a soft tide, pushing you to see deeper, to consider the hidden layers beneath what is visible. You notice the elegance of CRISPR's design, its dual nature as both guide and executor. It carries a map, a precise address, but also listens for context, for the subtle cues that ensure a cut is made in the right place at the right time. There is artistry here, a balance between precision and freedom. You imagine a sculptor shaping clay, feeling its texture and responding to its resistance. CRISPR moves similarly, shaping not with force, but with attentive touch, allowing the living material to participate in its own transformation. As the dance continues, you realize that this is more than a scientific technique. It is a philosophy, a way of approaching complexity with care. Each edit is an act of communication with life itself, a gentle suggestion rather than a demand. You feel a sense of kinship with the tool, as if it reflects a part of yourself, the desire to shape and improve, tempered by mindfulness and respect. The world of molecules and enzymes, once abstract and distant, now feels intimate, connected to the rhythm of your own thoughts and breath. The room around you seems to hum with quiet possibility. The DNA strands sway, responding to the movements of CRISPR, folding and unfolding like waves along a calm shore. You see patterns emerging, small ripples of change that hint at larger harmonies. Every adjustment carries potential, but also uncertainty, the reminder that even the most precise dance cannot predict every outcome. And yet, there is beauty in this uncertainty, a delicate tension between what is known and what is yet to unfold. You feel a gentle calm settle over you as you watch. CRISPR's touch is subtle, almost invisible, yet profound. It teaches patience, reminding you that change does not have to be abrupt to be meaningful. There is a quiet joy in observing the interplay of intention and response, of human insight and natural resilience. The molecules continue their rhythm, the DNA continues its spiraling dance, and you remain present, a silent witness to this intimate choreography of life. Thoughts of the broader world drift in. CRISPR is more than a tool, it is a symbol of what can be achieved when knowledge is wielded with care. You consider the potential for healing, for understanding, for creation without destruction. And yet, the questions linger, ethical, philosophical, and deeply human. How do we honor the balance of life while exploring its possibilities? How do we measure the impact of even the gentlest interventions? These questions hover softly, like clouds reflected on a still pond, inviting reflection rather than resolution. As the dance of CRISPR winds on, you sense the continuity of life around you. The molecules, the cells, the invisible threads of DNA, all are connected in a vast, unending network of interactions. You feel yourself part of this network, a participant in its unfolding story, even if only through observation and imagination. The gentle edits of CRISPR are a reminder that life can change, adapt, and heal, all while preserving its essential harmony. In the quiet of this molecular forest, you sense the rhythm of science and curiosity intertwined. CRISPR is not the end of the story, but a beginning, an opening into new ways of thinking, feeling, and understanding. You linger in the soft light, letting the delicate balance of precision and possibility wash over you. The whispers of DNA, the careful dance of CRISPR, and the echo of your own thoughts blend into a slow, meditative cadence. And as you slowly begin to drift from this molecular forest, you feel the pull of the next question, the next chapter in the journey of life, where the boundaries between what is known and what is imagined begin to blur, inviting you to wonder what comes after the dance. As you drift into the realm of the unseen, you find yourself in a world far smaller than a single grain of sand. Here, the air hums with an energy you cannot touch but can almost sense, a subtle vibration that carries the weight of life itself. Your journey leads you to a delicate lattice of spirals, loops, and twists, the DNA within each cell. It stretches endlessly before you, an intimate map of instructions, written in a language older than your own imagination. You move closer and notice figures flitting along these strands, moving with precision and grace. They are the guardians, the silent dancers whose mission is to protect the sanctity of your genetic code. Proteins and enzymes glide like dancers in a grand ballroom, their movements deliberate, almost lyrical. They do not speak in words you understand, yet their actions communicate stories of care, attention, and perseverance. Some pause to scan the double helix, their eyes, or perhaps what functions as eyes, skimming each base pair for errors. Others weave between strands, knitting together broken links, smoothing over disruptions that might otherwise ripple into chaos. You feel a quiet awe, watching these microscopic entities perform their nightly vigil, a choreography of repair and protection. A particular enzyme catches your attention. It moves slowly, deliberately, along a stretch of DNA that seems frayed at the edges. You notice how it pauses, leans closer, and with an almost imperceptible gesture, adjusts a misplaced piece. It reminds you of a gardener carefully pruning a delicate plant, removing what is wrong so that life can continue without interruption. You realize these dancers are not just workers, they are caretakers of continuity, ensuring that each instruction is preserved, each possibility remains open. You follow another figure, smaller, lighter, but no less important. This one carries a tiny cargo of repair materials, traveling with silent determination to a damaged site. As it reaches its destination, it pauses and carefully integrates its cargo into the existing structure. The DNA breathes around it, flexing gently, as if acknowledging the kindness of the intervention. Here, in this microscopic ballet, life seems soft and strong at the same time, a paradox embodied in motion. It is not always calm. Sometimes errors appear that are subtle yet insistent, mispaired bases that threaten to disrupt the delicate sequence. In those moments, multiple dancers converge, a coordinated ensemble responding to a single, fragile spot. They move with urgency, but never with panic, their energy focused and precise. The scene feels like a miniature symphony, each movement intentional, each correction a note in a song of survival. You recognize a pattern, a rhythm that resonates with something deep within you. It is the rhythm of care, of resilience, of life persisting through quiet diligence. As you watch, you begin to notice the individuality of each guardian. Some are elegant in their efficiency, moving with the flowing arcs of a practiced performer. Others are meticulous, almost ceremonious, performing every adjustment with deliberate reverence. And yet, all are united in purpose. You sense that there is no hierarchy here, only collaboration, a network of silent agreement that the work must continue. Each gesture, no matter how small, matters profoundly. The chromosomes themselves shimmer under your gaze, coiling and uncoiling like ribbons of light. You see how the dancers interact with these ribbons, sometimes spinning around them, sometimes gliding along them like a river current. They repair, they rearrange, they stabilize, but always with care, never with force. You imagine that if you could shrink down further, you might hear the soft hum of energy and connection, the faint resonance of life maintained through countless unseen acts of attention. You notice a pause in the dance. One guardian hesitates over a tiny imperfection, almost as if considering a choice. You realize that these guardians are not mechanical, they seem to possess a form of judgment, a sensitivity to the context of each correction. Perhaps this is why life persists, why errors do not always become disasters, why each cell carries within it the potential for both fragility and extraordinary resilience. Watching them, you feel a gentle reassurance, as if the body itself is whispering that it can tend to its own vulnerabilities. Time here is different. Seconds stretch and fold, moments seem both fleeting and eternal. You begin to feel that this is a lesson, quietly offered, that care does not demand speed, that vigilance need not be loud, that protection can be graceful and quiet. You think of the ways your own body silently mends, regenerates, and adapts, and you feel a soft wonder for the intricate, unending dance occurring inside you. As you move deeper, you see clusters of guardians, gathered around sections of DNA that are particularly active. They work in synchrony, a miniature crowd moving with purpose. You are struck by the poetry in this coordination. The idea that life, at its most fundamental level, is a conversation between molecules, a dialogue of maintenance and possibility. You feel privileged to witness such intimacy, to glimpse the elegance and devotion of processes that normally go unnoticed. Occasionally, mistakes persist despite intervention. You see a base pair that does not match perfectly, an anomaly that resists correction. Yet even here, the dancers do not despair, they adapt, shifting strategies, attempting gentle modifications, or simply accommodating the imperfection. You understand that resilience does not mean perfection. It means persistence, adaptation, and care extended over time. These dances embody patience, a reminder that life's continuity often relies more on gentle correction than on drastic change. As you begin to drift away from the DNA ballroom, you notice that the guardians do not vanish. They continue their vigil, a silent, eternal presence. You feel a quiet gratitude, sensing the invisible protection that underlies every heartbeat, every breath, every thought. You carry with you the image of their grace, their precision, and their unceasing dedication. It is a comfort, an assurance that life's fragility is met with equal and opposite resilience, and that within every cell lies a silent choreography of care. The last glimmers of the microscopic world fade, yet the sense of ongoing vigilance remains. You carry this awareness with you, a soft, meditative understanding that the body is not a passive vessel, but an active participant in its own story, repairing, adapting, and flourishing in ways both subtle and profound. And as you leave this hidden ballroom, your thoughts drift toward the next mystery, how these silent dancers communicate across distances, orchestrating harmony in the vast, invisible landscapes within, hinting at the next chapter of life's quiet, unending symphony. You begin your journey in a quiet place within yourself, where the hum of existence is soft and steady. Around you, the world feels suspended in gentle waves, a subtle rhythm echoing the pulse of life itself. And here, in this suspended calm, you sense the language of your own being, the intricate, delicate script of DNA. It is written in 4 letters, A, T, C, and G, simple in appearance, yet infinite in possibility. These letters are the brushstrokes of your biological story, each pairing with another to form the spiraling double helix, a staircase leading into the deepest corners of your identity. As you float closer, you notice that each sequence of letters is like a word in a language you have always known but never consciously read. The codons, 3-letter phrases that fold meaning into life's instructions, resonate like musical notes played in a gentle melody. You hear the rhythm of your genome, the tempo of creation that has been composed over billions of years. Some notes are soft, like whispers, others rise and fall in a deliberate cadence, like the ebb and flow of tides. You feel the music in your bones, and you understand that it is personal. Every rhythm, every pause, every note belongs to you. Through this melodic landscape, you explore the story of proteins emerging from the letters, as if words were forming objects and dreams simultaneously. Messenger RNA carries the instructions delicately, like a paper boat gliding down a silver river, and the ribosome reads each line, translating the language into the machinery of life. You watch, mesmerized, as amino acids join in a dance, folding into shapes both fragile and unbreakable, tiny sculptures animated with purpose. Each protein is a character in a quiet drama, playing its part in the symphony of your body, a reflection of the genetic whispers that guide all living beings. And then you notice the hidden poetry within the sequences. It is not merely information, it is dialogue. Your genome speaks to you in subtle ways, hinting at ancestral journeys, long-forgotten adaptations, and tiny choices made across millennia. You feel the presence of your grandparents in the twists of a strand, the echo of distant ancestors in the repetitive patterns that appear and fade. The chromosomes are like ancient scrolls, fragile yet resilient, holding within them stories of survival, curiosity, and wonder. Reading them, you are not simply observing, you are conversing with the quiet persistence of life itself. You wander through the interweaving paths of exons and introns, noticing how not all letters are meant to be read at once. Some are silences, pauses that give meaning to the sound, shaping the music without revealing themselves immediately. Others are enhancers, bright markers that amplify certain harmonies, guiding the story along its intended path. The interplay of activity and quiet creates a rhythm that is almost meditative, a pulse that mirrors the rise and fall of your own breathing. You realize that life is not a linear script but a layered poem, a dialogue of presence and absence, of expression and restraint. In this gentle exploration, mutations appear like subtle shifts in accent, small variations in phrasing that change the nuance without breaking the sentence. Some are soft, almost imperceptible, like a sigh in a quiet room. Others are more profound, altering the melody in ways that ripple through generations. Yet even these changes are part of the conversation, part of the dialogue that sustains life and allows it to adapt, to learn, to continue. You sense that each variation, whether inherited or new, is a note added to the eternal composition, a contribution to the symphony in which you are both listener and performer. You begin to see the wider tapestry, the network of connections that extend beyond your own cells. DNA is not isolated, it reaches outward in quiet universality. You touch the thread of a gene shared with other mammals, feeling a kinship that is both immediate and expansive. A sequence that once guided a tiny fin now guides your fingers, the same letters connecting the past to the present in subtle resonance. Every creature you pass in imagination carries echoes of the same melody, a shared rhythm that underlines the diversity of life with harmony rather than dissonance. Through this lens, you perceive the choreography of development. Each cell, as it divides and differentiates, reads a line from the book of life, responding to signals and contexts, balancing randomness with direction. You watch neurons branching like delicate trees, each connection forming the architecture of thought and sensation. You feel the quiet awe of watching this process unfold, the intimacy of witnessing life as it chooses pathways from a palette of genetic possibilities. The genome does not simply instruct, it converses, negotiates, and inspires, a living dialogue in which you are a participant as well as an observer. You notice the epigenetic notes, the subtle annotations that influence the reading of the script. Methyl groups and histones decorate the text, like punctuation added by invisible hands, shaping emphasis and intonation. These marks respond to the world, to experience, to nourishment, and to touch. The genome is alive not only in letters but in context, and you feel the gentle guidance of life responding to circumstance, adjusting, bending, but never breaking. You are part of this fluid conversation, feeling its subtle sway in your moods, in your growth, in the quiet unfolding of your day. As you drift further, you sense the infinite patience encoded in your DNA. Every replication, every repair, every interaction is a testament to resilience. The molecules themselves hum with quiet diligence, correcting errors with meticulous care, preserving continuity even in the face of chance. You understand that the genome is not merely a static book, it is a living manuscript, a work continuously edited by life itself, an ongoing dialogue that bridges the ephemeral and the eternal. You begin to notice the small, almost invisible patterns, palindromes, repeats, and motifs that recur like a refrain. These patterns form the rhythm beneath the narrative, a foundation for the visible structures. They are the hidden beats of the music, subtle yet essential, reminding you that much of life's wisdom is not shouted but whispered. You feel the gentle encouragement of the genome, guiding you through its lessons in patience, rhythm, and attention to the subtleties of being. In this meditative journey, the sense of time softens. The strands of DNA stretch beyond your perception, past your own life, past the lives of those you know, into a continuum of generations. You sense the quiet lineage of adaptation, the careful experimentation of evolution, the patient unfolding of complexity. And within this vast expanse, you recognize your place, not as a separate entity, but as a point of awareness within a living story that has been unfolding for billions of years. You feel the intimacy of this dialogue, the gentle trust it inspires. It is as if the genome itself is whispering secrets to you alone, offering insight without demand, revealing connections without judgment. Each sequence carries its history, its purpose, and its poetry. You understand that reading DNA is not merely a scientific act, it is an act of empathy, a communion with the processes that have shaped life and continue to shape it. And as you let the strands drift around you, the helix rising and falling like gentle waves, you sense the bridge to what comes next. The molecules will continue their dance, the sequences their dialogue, and the story of life its patient unfolding. And you, softly attentive, are ready to follow the next step, to see how this language transforms into the tangible structures and rhythms that make up the living world around you. You enter a quiet world beneath the surface of what you can see, where strings of life stretch like slender threads of light. Here, within the folds of cells, CRISPR moves not with clamor but with whispered purpose. Imagine it as a gentle visitor, a small feather gliding over a vast, intricate tapestry. It does not hurry, it pauses, hovers, senses the rhythm of the strands, and only touches when the moment feels exactly right. You watch the molecular scissors as they hover near the DNA, like a brush hovering above a canvas, waiting to make a single, intentional stroke. Each guide, each tiny piece of RNA, is a soft voice that tells the scissors where to land. There is no rush here, only a careful conversation between code and cutter, between possibility and responsibility. The world inside a cell is alive with motion, yet you notice its serenity. Molecules drift, collide, separate, and reform in a delicate ballet. CRISPR's movements are part of this dance, and the dance itself is part of a larger story of life unfolding. You sense that to intervene is not to dominate, but to listen deeply and to touch lightly. Every change CRISPR makes is deliberate. It is not about power, but about choice, a thoughtful pause, a gentle correction. Imagine a single note added to a long, flowing symphony, a note that changes the melody just enough to bring harmony without disruption. The science of cutting and editing is also the art of patience. You are aware of the responsibility that accompanies this ability. Even the tiniest shift in the code can ripple outward, affecting not only the cell but the organism, the ecosystem, the web of life that extends beyond your vision. As you breathe, you imagine the tiny molecules responding, almost like they recognize your care. There is mindfulness in this process, a deep ethical presence that asks you to consider consequences before action. Every cell becomes a teacher, showing you the balance between intervention and respect, between knowledge and humility. You begin to see CRISPR not just as a tool, but as a guide, teaching the human heart how to move with patience and care within the living world. Time stretches differently here. Minutes feel like centuries and yet pass in the blink of an eye. You watch as CRISPR slides along the DNA strand, scanning, sensing, searching. It pauses where the sequence matches the guide, a quiet acknowledgement, and then gently executes the cut. There is no violence in this, it is precise, mindful, necessary. The DNA responds, the cell repairs itself, and life continues, enriched by the gentle guidance rather than harmed by it. You consider the implications. To hold the ability to edit life is to hold a mirror to your own intentions. Each decision to intervene is weighted with the awareness of ripple effects. It is a meditation, a moment-by-moment practice of presence, where the hands of the scientist mirror the calm, attentive motion of CRISPR itself. You imagine your own mind slowing, becoming a vessel of patience and care, attuned to the subtle signals of life's ongoing conversation. In this quiet space, science feels like a meditation. The tools of biology are instruments of reflection, not just manipulation. You sense the echo of responsibility in each action. A single, precise cut here, a small adjustment there, a life touched lightly, thoughtfully, with awe. It is a lesson in restraint as much as power, a reminder that the ability to change the code of life comes paired with the need to honor its complexity. CRISPR reminds you that change need not be hurried. Sometimes the smallest, most delicate shift produces the most profound effect. You imagine a feather brushing away a single imperfection, a careful stroke that enhances the whole rather than erases it. Life, you realize, is a canvas too vast for force. It is a landscape that responds to gentle intention. The elegance of molecular design mirrors the quiet potential within your own choices, that you too may act with care, wisdom, and respect. And yet, even in this calm, there is a hint of tension. Life is unpredictable, resilient, and always evolving. Each edit, though precise, is part of an ongoing story whose end is not yet written. You sense a reverence for the unknown, a soft acknowledgement that while science grants knowledge, it does not remove mystery. You feel a quiet thrill in this, a recognition of the power and humility intertwined in creation itself. CRISPR teaches patience in motion. Watching the sequence align, the scissors hover, the cut made and repaired, you sense the rhythm of life. There is a harmony in this precision, a poetry in the way molecules respond, adapt, and restore. It is a lesson in stillness as much as in action, a gentle dance where intention meets natural flow. The tiny edits are a conversation with existence, a dialogue that whispers through each cell, handle this with care, and life will respond in kind. You notice that as much as CRISPR shapes DNA, it shapes perception. You become attuned to subtlety, noticing the spaces between actions, the pauses that allow life to breathe and grow. It is a meditation on effect and cause, on how even the smallest motion can carry meaning far beyond its immediate context. Each deliberate choice echoes through networks of life, connecting you to the vast, intricate patterns that stretch unseen beneath the surface. The dance continues, seamless and unhurried. Molecular feathers brush, strands align, corrections are made, and the cellular world hums along, guided yet undisturbed. You feel your own awareness deepen, attuned to the rhythm of living code and the quiet care embedded in each scientific gesture. You carry the sense that knowledge and responsibility are inseparable, that wisdom is measured in restraint as much as in skill. And as you linger in this quiet observation, you notice something else, a soft invitation to wonder. The more you watch, the more you see how life responds not merely to correction but to mindfulness, how intention and presence weave together with molecules to create harmony. You sense a bridge forming, between human curiosity and the natural order, between action and reflection, between the power to change and the humility to honor what is. You feel a gentle pull forward, an awareness that the dance of CRISPR is only one movement in the symphony of life. Beyond the precision, beyond the molecules, there is a larger question. How do we carry this knowledge in a way that is ethical, mindful, and caring? How do we step lightly into a world where each small choice ripples outward? You inhale this question, soft and open, like a slow tide washing over a quiet shore, and you realize that the journey is only beginning. And in this quiet, suspended moment, you drift toward the next understanding of how life repairs, renews, and surprises itself, carrying with you the soft, deliberate rhythm of the molecular dance. You begin your journey in the unseen garden of life, where sunlight barely touches, and yet the world hums with endless activity. Here, in the soil, on leaves, in droplets of water, billions of tiny beings carry stories older than any human memory. They are bacteria, each a universe unto itself, simple in shape, yet profound in wisdom. In their quiet existence, they endure attacks, navigate scarcity, and persist across generations, shaping ecosystems without any grand intention. Amidst this invisible bustle, you notice the invisible guardians. Within the microscopic genomes of bacteria lie arrays of repeating sequences, interspersed with fragments of viruses that once tried to invade. This is CRISPR, a library of remembered encounters, a diary of defense written in code. Every repeat, every spacer is a note, a memory of a confrontation survived, a lesson learned. You imagine the bacteria pausing, reading their own microscopic pages, readying themselves for the next challenge. The discovery of CRISPR begins not in a lab of bustling machinery, but in a patient gaze, in noticing patterns that others might have overlooked. Scientists, fueled by curiosity and quiet perseverance, observe these sequences. They see that these tiny beings, though lacking consciousness as we know it, carry tools of remarkable precision. Each fragment of viral DNA stored in the bacterial genome is like a beacon, a signal to recognize and neutralize threats. The elegance of this mechanism whispers to the human mind, even the smallest forms of life harbor solutions to problems we once thought insurmountable. As you walk through the narrative, you imagine yourself shrinking, falling into this microbial world, and seeing it from the bacteria's perspective. You watch as a virus approaches, a dark shadow moving swiftly through the aqueous landscape. The bacteria recognize the threat through the memory etched in their genome. They summon the Cas proteins, tiny molecular scissors that glide through the cell, searching, cutting, and disabling the invader with meticulous precision. It is a dance both fierce and graceful, survival encoded in movements smaller than the eye can perceive. Human minds, observing this dance, begin to wonder, what if we could guide these molecular scissors? Could we, too, write with this language, editing the strands of life itself? Here begins a gentle merging of curiosity and creativity. CRISPR is no longer just a bacterial defense, it becomes a bridge. The wisdom of millennia condensed into sequences, once hidden in invisible chambers, now offers a hand to human imagination. The realization is humbling: to edit life, one must first learn to listen. You feel the pulse of patience required, the slow, steady rhythm of discovery. The first experiments are tentative. Scientists speak to the bacterial language carefully, testing the limits of understanding. A fragment of DNA here, a guide RNA there, and slowly, the possibilities unfold. It is not instantaneous mastery but gradual learning, much like tending a garden. Mistakes are lessons, failures are whispers of what the system refuses, what is not yet understood. In these moments, science becomes almost a meditation, a dialogue with the subtle, persistent intelligence of nature. The narrative of CRISPR is interwoven with stories of curiosity, persistence, and wonder. You imagine the early researchers, peering through microscopes, scribbling notes in dimly lit rooms, captivated by the invisible choreography within each cell. They follow patterns, trace sequences, and slowly understand the architecture of microbial memory. Each step feels deliberate, rhythmic, almost like listening to a slow, unfolding melody. There is joy in small discoveries, a spacer identified, a protein function confirmed, a mechanism clarified. Each moment is a quiet triumph, a testament to patience and attentiveness. In this world of microscopic guardians, you sense lessons beyond biology. The bacteria show resilience, the ability to remember, respond, and adapt. They illustrate innovation at the most fundamental level. Solutions arise from constraints, creativity from necessity. And you, wandering through this narrative, recognize a parallel in human life. Challenges are met not by brute force, but by attentive observation, by listening carefully to what has come before, by crafting responses with precision and care. CRISPR becomes a metaphor, a bridge connecting the wisdom of small, patient systems to our own larger, often chaotic world. You imagine guiding a CRISPR complex as if holding a delicate brush. Each cut, each edit, requires awareness and gentleness. The excitement is tempered by respect for complexity, a recognition that even the smallest error can ripple outward. The bacterial model offers both a tool and a cautionary tale. Mastery is not just about capability, but also about humility and attentiveness. Science, you realize, is not a conquest, but a conversation with the intricacies of life. As the chapter unfolds, you see the impact of these discoveries ripple outward. CRISPR moves from lab benches to medical hope. The once invisible guardians of microbial life now illuminate paths toward healing. Therapies for genetic disorders, innovations in agriculture, experiments in ecological restoration. Yet, the story retains its quiet wonder. These applications are not framed as triumphs over nature, but as collaborations with its inherent intelligence. You sense a reverence threaded through each experiment, a careful negotiation with life's code. The narrative invites reflection on time and scale. The memory encoded in bacterial DNA spans generations, while human understanding is fleeting yet intensely focused. There is poetry in this intersection, ancient microbial strategies meeting the bright, transient curiosity of human thought. CRISPR, in essence, is a bridge across eons, a testament to the patience of life and the persistent reach of imagination. You pause, imagining the billions of interactions, the countless tiny battles, each one contributing to a mosaic of survival, each one quietly shaping the future. Your journey through this chapter lingers on the notion of listening. Not the passive hearing of sound, but the active, contemplative tuning into patterns, signals, and the subtle cues life provides. The bacteria, in their simplicity, teach you to attend, to notice, to respond with care. They remind you that knowledge is not merely accumulation, but intimacy, understanding the rhythms of a system, the memory it carries, the potential it holds. The discovery of CRISPR is thus a story not just of science, but of relationship, attention, and the unfolding of understanding. You feel the chapter winding into its reflective cadence. The bacterial world fades gently from view, leaving impressions of order, persistence, and subtle elegance. The molecular dances you witnessed are not lost, they echo quietly in your mind. You are left with a sense of awe at the delicate balance of life, the ingenuity encoded in simplicity, and the profound potential unlocked when human curiosity listens and learns. In the quiet of this moment, you recognize the invitation to explore further, to question more deeply, to journey toward the next revelation that awaits beyond the visible. And as the soft glow of awareness settles around you, a whisper of thought emerges, a question for the next chapter, a gentle nudge toward the story of how these discoveries unfold into the human world, touching life in ways both subtle and transformative, and asking whether we are ready to meet the responsibility that comes with such intimate knowledge. You begin your journey at the edge of a quiet forest, where light filters softly through leaves that seem to shimmer in slow, deliberate waves. Each leaf carries its own story, a history written in lines too small for eyes to see, yet crucial to the forest's health. In this forest, there are threads of life, unseen but undeniable, weaving between roots, whispering across moss, curling around the delicate veins of each plant. These threads are the code of life itself, the DNA, the quiet blueprint of every living thing, and they hold the potential for both harm and healing. Imagine a single thread that is frayed, twisted in a way that the forest cannot fully flourish. The creatures that rely on its pattern falter, the ecosystem murmurs in uncertainty, and yet the thread itself waits patiently, unaware of the imperfection. Here is where CRISPR steps lightly, a gentle caretaker, a pair of skilled hands that do not tear or rush, but observe and correct. You can see it as a painter adjusting a brushstroke on a vast, living canvas, or as sunlight nudging the shadows to soften, illuminating hidden corners without disturbing the natural rhythm. CRISPR is a tool, precise and deliberate, guided by an understanding of what the thread should be. It seeks a matching sequence, a companion that whispers, "This is where change is needed." It moves along the spirals of DNA, scanning with an elegant attention that mirrors the patience of the forest itself. In your mind, you can almost hear a soft hum as the molecule locates its target, a whisper among the leaves, a quiet understanding between caretaker and forest. There is no hurry, no clamor, only the gentle negotiation of life-seeking balance. As you watch, you see CRISPR's tiny molecular scissors at work. They do not cut recklessly, they puncture only where there is a known imperfection, opening a space for the thread to mend itself. It is as if the forest has paused to allow sunlight to illuminate the exact spot where moss must grow, where roots must intertwine. And then, in a silent miracle, the natural repair mechanisms of life do their work, weaving the thread back into harmony. There is elegance in this simplicity. The forest does not fight the intervention, nor does it collapse under its own fragility. It merely continues, stronger, more complete, a testament to quiet restoration. You take a deeper breath and notice how this mirrors life beyond the forest. Each of us carries unseen threads, imperceptible patterns that shape who we are, how we respond, and what we inherit. In CRISPR's gentle dance, there is a metaphor for human care, the way we might correct what is broken, the way we might help one another flourish, not through force but through understanding. It is not about controlling, but about listening and responding with precision and empathy. You feel a sense of peace, realizing that science, at its most refined, is a form of listening, a way to hear the quiet language of life itself. The beauty of this process is its subtlety. You imagine a single strand of DNA, composed of a sequence of letters, carrying instructions for a tiny protein somewhere in the cells of an organism. One letter out of place can ripple outward, affecting countless systems. But when CRISPR corrects that letter, the effect is not explosive, it is a soft restoration, a return to rhythm. The protein functions as it was always intended, and the organism continues, unaware of the invisible correction, yet carrying the benefit in every breath, every movement, every heartbeat. You wander deeper into the forest, tracing the pathways of light as it dances across the ground. You realize that CRISPR is not magic, it is intention made tangible. It requires knowledge, patience, and humility. The forest does not yield its secrets easily, and neither does life itself. But when approached with care, each correction is a quiet act of stewardship, a restoration that honors the complexity of what it touches. There is no need for grand gestures or dramatic interventions. The smallest, most precise action can ripple across an entire system. In your mind's eye, you see countless threads, each one unique, fragile, resilient. Some have imperfections that are subtle, almost invisible, yet they affect the harmony of the whole. CRISPR allows us to navigate these imperfections with care. It is a reminder that science, at its best, is not conquest but caretaking, not domination but dialogue with the living world. You feel a gentle awe at this, a respect for the meticulous work happening at scales invisible to human eyes, a reverence for the way life maintains balance when guided with understanding. You begin to notice patterns in the forest that you had missed before, the way the light bends through leaves to touch the forest floor, the way roots interlace beneath your feet, the gentle sway of the trees in response to a hidden current of air. Each movement is both a response and a preparation, a delicate negotiation that keeps the system alive. This is much like CRISPR in action, a subtle intervention that harmonizes with natural processes rather than imposing upon them. The more you observe, the more you sense the quiet intelligence of life, and the more you understand that correction and care are inseparable from growth and survival. CRISPR's role is not to create perfection where none exists, but to facilitate potential. It does not insist on rigidity, it does not demand uniformity. Instead, it enables what is already present, restoring pathways that have faltered, helping systems regain their natural rhythm. There is humility in this approach, a recognition that life knows how to heal itself when given guidance, and that the human hand must move with reverence and attentiveness. You feel a deep calm, a soft assurance that even the smallest, most invisible intervention can have profound effects when executed with respect and precision. As you walk along a stream that threads through the forest, you notice how the water adapts to every stone, every root, every subtle slope. It flows around obstacles, reshaping its path with patience and persistence. This flow is akin to the molecular choreography of CRISPR, adjusting, correcting, yet always following the inherent logic of the system it touches. There is grace here, a reminder that intervention need not be violent or disruptive. True healing, whether in nature or within living cells, moves with rhythm, timing, and sensitivity. You feel this understanding settle into you like sunlight warming a cool stone, and you carry the calm of it as you move through the forest of your thoughts. And so, you begin to see CRISPR as a storyteller, telling the tale of life's quiet resilience, its capacity for repair, and its endless potential for renewal. Every correction is a sentence in an ongoing narrative, each gently guiding the plot without erasing the past. You imagine the threads of DNA as lines of poetry, each letter a note in a larger symphony, and CRISPR as a soft editor, ensuring the music plays without discord. There is no rush, no applause, the change is quiet, profound, and lasting. In this moment, you understand that the essence of CRISPR's work is trust in life's own mechanisms, trust in the delicate balance of the unseen threads, trust in the slow unfolding of possibility. You feel a kinship with every cell it touches, every organism it helps restore, every sequence it corrects. The forest around you seems to breathe with you, its shadows and light reflecting the gentle negotiation of repair and renewal. And as you follow the sun-dappled paths deeper into the woods, you know that this is only the beginning of understanding, that each restored thread opens new questions, new pathways, new glimpses into the quiet architecture of life itself. You pause by a clearing where the forest seems to exhale, the air thick with the scent of moss and stone. You realize that each intervention, each correction, is an invitation to look closer, to observe more patiently, to engage with life in a manner that honors both fragility and strength. CRISPR moves with grace because it respects what it touches. And in that respect, there is beauty, there is hope, there is a deep, unspoken connection between science and the life it seeks to nurture. The forest seems to whisper as you continue walking, inviting you to consider the next stage of this quiet journey, the possibilities that unfold when knowledge meets care, when precision meets patience. You feel a gentle anticipation, a sense of soft wonder, knowing that the path of understanding is ongoing, and that each small act of restoration is part of a greater, unfolding story that stretches far beyond the edges of your sight. You step quietly into a world smaller than the eye can see, where threads of light curl and spiral in endless patterns. Each strand of DNA rests within its microscopic landscape, like a riverbed frozen in perfect motion. You imagine the molecules as dancers, moving with grace that is invisible but deeply felt. Their rhythm is not loud, it does not shout or demand attention. Instead, it hums softly, a gentle vibration that resonates in the quiet spaces of your mind. You are a silent observer, watching as these dancers twirl and bow, each motion carrying the memory of generations. The way DNA coils and stretches reminds you of ribbons floating in a slow current, each twist purposeful yet free. Within these delicate spirals lies the story of life itself, a story written not in words but in chemical whispers. The letters of this code, 4 in number yet infinite in combinations, form the instructions for every heartbeat, every breath, every flicker of light across a leaf. You feel the weight and the wonder of it all, the vastness contained within such minute forms. It is as if you have entered a cathedral built of possibility, where every corner holds a secret and every shadow a new revelation. You notice the subtle changes, the edits, that can be made to this intricate tapestry. They are almost like gentle nudges in a dance, a step altered, a movement extended, a rhythm shifted. CRISPR, the tool that allows such precise editing, is a hand guiding a brush across a canvas, careful not to disturb the natural flow, yet leaving a mark that can alter the image entirely. Each cut into the DNA is small, almost imperceptible, but its effects ripple outward in ways both immediate and far-reaching. As you watch, you imagine the ripples spreading across the pond of life. One tiny change in a single cell may echo through generations, touching forms yet unborn, shaping the dance of life in ways no one can fully predict. There is a strange comfort in this, a sense that even the smallest touch can carry purpose. And yet, there is also responsibility. Each choice, each intervention, is paired with an awareness that life's code is not yours alone to command. You are part of a continuum, a lineage of observers and participants, connected to the past and the future by these delicate, swirling threads. The molecules respond to edits with a quiet elegance. A missing letter may cause a ripple, a slight pause in the choreography, a new step appearing where none existed before. You notice the balance between chaos and order, between chance and design. Life itself is not rigid, it is a flexible song, with room for improvisation within a framework of rules. And in this flexibility lies potential, a deep and soft promise that change, when guided with care, can blossom into beauty. You feel a gentle awe at the realization that you are witnessing the very essence of adaptability, evolution unfolding in miniature before your eyes. The cells are alive with a subtle energy, a luminous dance of creation and repair. Proteins fold and unfold like origami in slow motion. Enzymes glide along strands of DNA like skaters across an invisible rink. You sense their purpose, their quiet persistence. Each action is both precise and tender, a testament to the intelligence embedded in the machinery of life. It is a choreography you cannot rush, one that teaches patience as much as curiosity. Here, you understand that every cut, every correction, must be approached with both reverence and mindfulness. As you drift further into this cellular world, you reflect on the ethical questions that whisper through your thoughts. The ability to edit life is both a gift and a responsibility. How do you decide which dances to alter, which movements to leave untouched? The question is not urgent, it does not demand an answer immediately. Instead, it floats gently around you, a soft echo that encourages reflection. You understand that curiosity and care must travel together, that wonder without responsibility is incomplete, and responsibility without imagination can become rigid. You notice that every edited strand carries a story, an unspoken dialogue between past and future. A correction in a gene associated with disease might prevent suffering for generations to come, like smoothing a path across a rocky landscape. Yet each action also reshapes the environment in ways subtle and unseen, like ripples moving outward to touch shores far away. The consequences are not always visible, not always immediate. You feel a patient awareness growing within you, a willingness to sit with uncertainty and observe before intervening. Time seems to stretch and bend in this quiet world. You are aware that within the nucleus of a single cell, billions of reactions occur every second, yet the pace feels unhurried. The edits, the repairs, the dances of proteins and enzymes, all flow with a rhythm that is both constant and gentle. You feel as if you are listening to the heartbeat of life itself, steady, resilient, and ever-changing. There is a meditative quality here, a soft understanding that life is both delicate and enduring, fragile yet incredibly robust. You are drawn to the notion of inheritance, the way changes echo across generations. Each decision you imagine as part of this narrative feels like a ripple in a calm pond, small and quiet yet inevitable in its reach. You picture the descendants of cells, each carrying traces of your imagined touch, moving forward into the unknown. There is no urgency, no need to control every outcome. Instead, there is reverence for the complexity, for the interwoven relationships that define life's choreography. You sense that even the smallest gesture can resonate with meaning, and that patience is as important as precision. The dancers of DNA seem aware of your presence, not in a literal way, but as if your consciousness aligns with their rhythm. You notice the way strands fold and loop, the way enzymes approach with care, the elegance of a well-timed correction. Each action is intentional, guided by rules encoded in the molecular machinery, yet imbued with a grace that feels almost conscious. You are reminded that intelligence exists not only in thought but in pattern, in interaction, in the delicate balance of forces. Watching this, you feel a deep connection to the ongoing story of life, a story that you are part of simply by being a witness. As the chapter unfolds in your mind, you understand that change is both gentle and powerful. Every edit in DNA is a quiet act of creation, a decision made in the context of a vast, interconnected system. You feel the weight of possibility, tempered by awe and respect. The molecules do not rush, they do not question your presence. They move with an inherent wisdom, carrying forward the legacy of countless generations, a rhythm that continues regardless of observation yet feels enhanced by it. You let yourself linger in this quiet reflection, absorbing the profound calm that comes from understanding life's subtle complexity. There is no need for immediate answers, no demand to act. Instead, you are invited to breathe in the elegance of molecular dance, to feel the reverberations of your curiosity ripple outward, and to honor the responsibility that comes with knowledge. You sense that awareness itself is a kind of care, a soft engagement with the intricate choreography of existence. And as you prepare to leave this miniature cathedral of motion, you notice a new question forming at the edge of your thoughts. How will these ripples carry forward, shaping the unseen future of life? The dancers of DNA continue their timeless motions, indifferent yet intimate, leaving you with a lingering sense of wonder and gentle anticipation. You step back slowly, carrying the memory of their rhythm with you, knowing that the next chapter of exploration awaits, a continuation of curiosity, responsibility, and the quiet beauty of life's unfolding story. You begin in a quiet room, where the soft hum of existence surrounds you like a gentle tide. Outside, the night sky stretches wide, an unbroken canvas of possibility. Each star is a question, a choice, a path that bends in ways only the cosmos can understand. You feel your own heartbeat echoing the rhythm of the universe, small yet conscious, fragile yet capable. In this delicate silence, you sense that science is not merely observation, it is dialogue, a conversation whispered across the expanse of time and space. As you breathe slowly, the stars seem to shimmer in response, their light a guide to decisions that weigh on your mind. Ethics appear here, in the luminous gaps between what is possible and what is wise. You imagine them as gentle constellations, soft lines connecting distant points of light, creating shapes that suggest balance, harmony, restraint. They do not demand action, they invite reflection. Each choice you consider now feels like stepping lightly across an open sky, aware of the fragile equilibrium beneath your feet. You picture a single atom, suspended in the quiet of your imagination. It spins in place, a universe unto itself. You think of the power it holds, the potential for creation or disruption. The laws that govern it are strict yet elegant, offering a pattern that is at once predictable and mysterious. In the same way, your choices carry weight. The tiniest shift in thought, the gentlest application of wisdom, can ripple outward, touching lives and worlds in ways unseen. The connection between ethics and science becomes a soft, humming current, guiding your gaze back to the stars above. The moon hangs low, a pale sentinel observing the interplay of potential and restraint. You notice how light falls across its surface, illuminating craters that tell stories of collision and time. Each mark is a reminder that even in forces beyond control, there is history, memory, and consequence. You recognize that your own actions, too, leave traces, subtle yet enduring. Ethics, in this sense, are the moonlight in your mind, a reflection of possibilities tempered by awareness. You feel your body relax, the weight of indecision softening into contemplative calm. A gentle breeze moves through the open window, carrying with it the scent of earth, of growth, of change. You close your eyes for a moment, imagining the way choices might unfold. A decision, though small, can spiral into effects both wondrous and profound. You see scientists, past and present, navigating this delicate landscape, their curiosity tempered by foresight, their imagination guided by care. You understand that the pursuit of knowledge is inseparable from the responsibility to wield it thoughtfully. The quiet of the night allows this truth to settle softly within you, like a star sinking gently below the horizon yet leaving its glow behind. You turn your attention inward, to the pulse of understanding that lies beneath thought. Every question you ask of the universe carries with it ethical weight. How will your discoveries be used? Who benefits? Who bears the cost? These inquiries do not diminish wonder, they enrich it. Ethics become not a restriction, but a lens through which the beauty of science is fully appreciated. You imagine a stream of silver light flowing from each decision, connecting you to the world around you, to other minds, to other possibilities yet unseen. Each reflection is a ripple on this river of understanding, subtle and enduring. In the stillness, time seems to stretch. You move among imagined laboratories, watching researchers handle fragile instruments, like caretakers of a sacred trust. Their hands hover over microscopes, pipettes, and delicate slides, not in haste, but with intention. You sense the reverence in each careful motion, the unspoken dialogue with knowledge and consequence. Choices, here, are not made lightly. They echo the same rhythm you feel in your own heart, a rhythm that balances curiosity with compassion, power with restraint. Science becomes a meditation, and your thoughts move with it, slow, deliberate, and luminous. You imagine the night sky as a metaphor for your own mind. Nebulae swirl softly, clouds of possibility yet unformed. The galaxies spin in silent harmony, each movement governed by invisible laws. In this vastness, decisions are not solitary, they exist in relation to everything around them. You sense that ethics are the invisible threads connecting one star to another, one action to another, one being to another. The universe does not demand perfection, only mindfulness. Each reflection, each pause, becomes a step in the dance of understanding, a gentle sway in the endless rhythm of thought and consequence. A soft voice seems to whisper within you, the echo of curiosity shaped by care. You imagine being part of a continuum of minds, past and future, each contributing to the conversation of the cosmos. You see that restraint is a form of wisdom, that compassion guides innovation, that power is best wielded lightly. And as you watch the stars flicker in distant constellations, you feel a profound sense of responsibility, coupled with awe. Knowledge and morality are inseparable companions, twin lights guiding you across the vast, silent expanse of possibility. Your gaze drifts toward a cluster of stars, brighter than the rest, and you allow yourself to imagine the choices that await. Not all decisions are urgent, some can linger in thought, waiting for the right moment to take shape. You feel your own mind as a calm observatory, where each idea and concern has space to breathe. In this quiet, the connection between science and ethics becomes a soft rhythm, a lullaby of contemplation. You sense that understanding is not a destination, but a gentle journey, unfolding one reflective step at a time. And so, as you lie beneath this imagined sky, you sense the soft weight of possibilities yet to come. Ethics, like distant stars, do not illuminate every path completely, but they offer a quiet, guiding glow. You allow your mind to rest in this luminous space, aware of the choices ahead, the responsibilities carried gently in thought and intention. The night does not end, it simply continues, a horizon of inquiry that invites you onward. The universe waits patiently, and you feel the calm, deliberate pulse of science and conscience entwined, guiding you softly toward what comes next. You close your eyes, and the world around you softens. The sounds of your day, distant traffic, whispers of wind, the faint hum of your thoughts, melt into quiet. In this gentle quiet, your mind turns inward, and there, within the calm, you begin to imagine the threads of life itself, the delicate spirals of DNA. They stretch endlessly, twisting and curling like smoke rising from a candle. Each turn, each fold, is precise yet fluid, a dance of microscopic elegance. You watch these strands move, their rhythms slow and hypnotic, as if time itself has softened to match the pace of your breath. Imagine a single cell. Within its tiny walls lies the vast library of life, every possibility encoded in letters smaller than your eyelash. And there, nestled among these letters, is a story of intervention, of gentle guidance, CRISPR. You see it not as a tool, cold and mechanical, but as a quiet, purposeful presence moving through the labyrinth of DNA. It glides with care, seeking the right place to pause, to nudge, to suggest a tiny correction. Like a painter delicately tracing a line in the sand, it does not rush, it does not disturb, it simply helps life refine its own poem. The spirals bend and shimmer, responding to this invisible touch. In your mind, CRISPR appears as a soft light, a wisp of understanding moving along the double helix, pausing where the story calls for it. And here, in this serene observation, you realize that editing DNA is more than changing letters. It is conversation. It is listening to the hum of life and offering a whisper of possibility. You sense the patience required, the ethics, the tender consideration. CRISPR, in this quiet narrative, becomes less a technology and more a companion to life, walking alongside it with respect. You drift further, and the scene expands. Each gene is no longer just a sequence of letters, it is a thread in an immense tapestry, shimmering with color you cannot name. When CRISPR intervenes, it does not tear. It does not impose. Instead, it subtly weaves new patterns into the existing fabric. The threads hum with new potential, gentle and unforced, as if the universe itself were holding its breath, curious about this small, precise change. You feel a soft excitement here, a warm awe, but it is the quiet kind that lingers, the kind that does not startle the mind or pull at the heart too sharply. Consider the act itself, the cutting, the guiding, the repair. These are not violent acts in your imagination, they are gestures, tender and deliberate. A tiny enzyme, Cas9, moves like a shadow, finding its target with a focus that seems almost meditative. It does not hurry. It pauses, recognizes, and then guides the cell to the next step. You can almost hear the faint echo of molecular conversation, a gentle suggestion, a consented change, an agreement to evolve. Life, in your mind's eye, does not resist. It responds, quietly, with subtle elegance. Now, let your awareness drift outward, beyond the microscopic. You begin to see the implications, the whispers of possibility that ripple from each small change. A plant, once struggling, now flourishes. A bacterium, once rigid, now adapts to a new rhythm. The choices that life can make, with CRISPR as a careful guide, are countless, yet they do not feel overwhelming. They feel like pebbles dropped into a still pond, each sending concentric waves of gentle wonder across the surface of understanding. You watch the ripples expand slowly, hypnotically, knowing that you are witnessing the quiet poetry of evolution's hand, now accompanied by human insight. Your breath lengthens as you sink deeper into this vision. Each inhalation carries the warmth of comprehension, each exhalation releases the tension of complexity. The double helix continues its graceful twists, and you sense that CRISPR is not altering life recklessly. It is suggesting, exploring, opening doors that were once firmly closed. It is the gentle curiosity of a child peering into a garden at dusk, careful not to crush the flowers, careful not to wake the bees. In this soft light, you understand that science, at its best, can be a lullaby, a story that invites the mind to wander safely, to imagine, to rest. The journey takes you next to the microscopic theater where life unfolds. You see molecules moving like dancers across an invisible stage, enzymes guiding, nucleotides responding. The choreography is ancient and intricate, yet here, in your mind's gentle vision, it becomes accessible. You follow a single act, a cut, a repair, a sequence replaced. You do not need to rush through it, there is no urgency. Instead, you savor each moment as if it were a note in a lullaby, each molecular gesture resonating with quiet intelligence. Even the errors, the pauses, the hesitations, feel like soft punctuation in a story that is still unfolding. As the scene widens again, you realize that you are floating, gently, over a vast landscape of living possibilities. Here, CRISPR is not just a tool but a philosophy, an approach to life that honors the smallest components while opening doors to new harmony. You see how this delicate dance reflects your own inner rhythm, curiosity tempered by care, understanding balanced with humility. In this space, science and meditation merge. You are both observer and participant, aware of the grandeur of life, yet cradled in its intimate details. You understand, in a subtle way, that to watch is also to honor. The final vision of the chapter draws you into a twilight of thought. You watch the spirals of DNA continue their endless, graceful motion. CRISPR rests for a moment, a pause that is almost reverent. The double helix shimmers, reflecting the faint glow of understanding you carry within you. You feel the quiet assurance that these tiny interventions, guided by knowledge and care, can ripple outward in beauty and possibility. And as your mind drifts closer to sleep, you sense that each moment of curiosity, each gentle experiment, is not a disturbance but a conversation, one that invites wonder rather than fear. Now, as you begin to sink fully into slumber, the soft hum of life continues around you, around your imagination. DNA spirals, CRISPR guides, molecules dance, and you, too, float in the calm awareness of possibility. You carry with you a sense of gentle empowerment, a recognition that even in the smallest acts of understanding, there is a bridge between curiosity and care. And as your thoughts begin to quiet, the whisper of the next chapter begins to emerge, a soft invitation to explore not just the mechanics, but the ethics, the choices, the tender responsibility that accompanies the power to guide life itself.